Hi, my name is Amelia Braun, and thank you for coming to this presentation on my SHORE work involving the morphological and physiological variations associated with reproductive and vegetative salinity tolerance in sorghum. As the global population continues to climb, there is a need to increase food production. However, climate change through temperature increases and adverse weather patterns can make agriculture extremely difficult. Salinity stress in particular is becoming a serious problem, especially affecting areas such as Northern Africa, the Middle East and parts of Central Asia, and Australia, as can be seen in Figure 1. My project aims to study the impacts of salinity stress on the vegetative and reproductive traits of sorghum, which is a popular cereal grain cultivated and eaten in Africa and Asia. My work may help create a better understanding for future salt tolerance research, as well as help identify varieties that can be used for agriculture on these salt-contaminated lands. To achieve this, I selected 25 different sorghum accessions, each with varied known salt tolerance responses, and I'm using these to investigate the relationship between vegetative and reproductive tolerance. This means I will be analyzing how the plants will respond to salinity stress through both their growth and overall vegetation, as well as through their grain production and quality. These were divided into either a control group, which is watered with tap water, or a treatment group, which is watered with 75 millimolar NaCl solution. A variety of vegetative measurements are being measured throughout, including the height, the cone diameter, leaf chlorophyll, and leaf temperature. The project will continue until the plants flower and their seed reaches maturation, at which case the final measurements of biomass, grain yield, and seed color, diameter, and weight will be measured. Figure 4 shows sorghum's phylogenetic tree as well as known salt tolerance values from Henderson et al. Yellow indicates salt sensitivity, while red indicates salt tolerance. In general, genotypes towards the base of the tree tend to have higher tolerance values, while along the evolutionary line, sensitivity tends to increase. This is potentially because as sorghum has been continually domesticated and improved, this trait has been more or less lost. Prior to experiment start, the germplasm were measured for their seed diameter, color, and weight. Figure 5 shows a non-metric multidimensional scaling test and associated analysis of similarity, in which there was no significant clustering of these traits in regard to the expected tolerance values from Figure 4. This means that in the germplasm, these traits have no relationship to the expected response under salinity stress. However, though not significant, there does appear to be mild clustering of some of the tolerant accessions circled in red on the graph. This may potentially indicate that post-salt exposure, these accessions will be significantly clustered in regards to seed weight, diameter, and color. Though this research is ongoing, the data I have collected so far indicates that the germplasm, along with being a poor indicator of predicted tolerance, also has no relationship to domestication and improvement. This is shown by Tukey-Kramer honestly significance difference test, in which there was no pattern regarding either the germplasm diameter or the color based on the known tolerance values or the position on the phylogenetic tree from Figure 4. In addition, one-way ANOVAs were performed on the height and cone diameter data. Only 4 of the accessions showed significantly reduced height under treatment conditions, and only 1 of the accessions showed a significantly thinner cone as compared to controls. Because these accessions came from a variety of locations along the phylogenetic tree, as well as had a variety of salt tolerance responses, with 2 of them being tolerant, 1 of them being sensitive, and 1 of them being intermediate tolerance, I concluded that pre-flowering, salinity stress has little impact on sorghum's morphology. Figure 6 shows the comparison between a control and a treatment group of the one accession that was significant in regards to both height and culm diameter. I would like to thank my capstone committee, Hawkins Lab, the SURE program, and the Honors Excel program and Henry Hurlbut Memorial Research Fund for funding this experiment. Thank you for listening to my presentation.